And the live is going in a few seconds. You're live.
Okay. So we move to keep it a little bit up. So you give me time?
And the live is going in a few seconds. You're live.
So welcome everybody to this special meeting of the Faculty of Science. We are here together today for the public defense of the PhD of Justin Tiano in the Faculty of Science, Marine Science. So Justin will have now 30 to 40 minutes to present his thesis, and after the presentation we will give the members of the jury the opportunity to pose their questions. If there are questions from the audience, we can take them as well, or maybe even through the chat. So Justin, please, you have the floor now for about 30 to 40 minutes.
Okay. All right, so thank you all for being here today. Happy to have you here either in person or joining online. Definitely happy that we could get that going. So let's get things started. Electric pulse trawling. Is it really as bad as everyone says, or is it actually a good alternative? Now, our research is so much more than just trying to see if any method is good or bad, but throughout my PhD, I haven't really been able to escape this question. And so what I hope for all of you is at the end of this talk, you'll all be able to answer this question yourselves, given your own perspective on the topic. I'll be giving a summary today of my PhD, which is titled Evaluating the Consequences of Bottom Trawling on Benthic Pelagic Coupling and Ecosystem Function. And so basically, we're taking a look at how bottom trawls affect biogeochemistry, which is still an understudied topic in fisheries science. So it involves things like changes to benthic nutrients and organic matter, but also changes to the benthic invertebrates that have such a strong effect on these different dynamics. So much of my research in the past few years has been looking at this controversial pulse trawl topic. So this has been in the news a lot in the past few years. It's been voted to be banned in the EU, and it's currently being phased out. But regardless of whatever happens politically, our research remains very relevant. And this is because it's applicable to a lot more than just pulse trawling. I mean, really, we could apply this work to whatever is scraping along the top of the seabed. And I'm sure a lot of people who are listening in are curious still to see if this ban was really justified. And now, to be clear, it is not my goal to actually say that it was or that it wasn't, but perhaps some of this research can shed some light on this topic. So now there's this thought that the French and English are against pulse trawling and the Dutch are for pulse trawling. Now I'm actually American, but this still kind of puts me in a bit of an awkward situation because my wife is French, so I can't really be anti-French. I also live and work in the Netherlands, and so it's difficult for me to be anti-Dutch as well. And so naturally, it puts me in a good position to stay neutral on this topic, as I should. And people say things all the time, like the Dutch versus the French argument, and it's really much more complicated than this. And but you hear it all the time. Now, in all seriousness, our objective throughout this research was to steer clear from all this politics and just stick to the science in order to remain as unbiased as we possibly could and answer our given research questions, which starts out with what is the effect of pulse trawling on the functioning and biogeochemistry of benthic ecosystems, and this stems from the original research proposal. But in order to put things into context, we really need to compare it to its alternative, which is conventional tickler chain beam trawls. Now, I do almost as much research into beam trawling as I do with pulse trawling. Now, also in Chapter 4, we do an experimental study where we try to look at the direct effects of electricity itself, and also we look at the effects of sediment resuspension and just physically disturbing the top of the sediment. And so I start off in the first chapter by setting the scene a little bit and talking about the North Sea. It's one of the busiest bodies of water in the world. It's been trawled for hundreds of years, probably intensively for about 100 years. And so it's really difficult to find a pristine area, an untouched area of the North Sea to use as a reference point. And so a lot of these places that we call controls or so-called untrawled areas are only relatively untrawled. And really they actually have a history of trawling before that, but we can't really avoid this. And it's good to understand that. I also spend the rest of the chapter reviewing the literature on bottom trawling effects on benthic communities and biogeochemistry. But first, I'd like to talk about the gears themselves, which I do the most research on. And so, on the left here, we have a standard tickler chain beam trawler. These use these chains to kind of dig up the fish from, flatfish from the bottom of the sediment, and they're then easily caught in these nets which drag along the seafloor. Now, I have to be specific here. On the right-hand side, we have a type of flatfish pulse trawler. Now, my research is mostly on flatfish pulse trawling, and I have to be specific about this because there's also pulse trawling for shrimp, and in Scotland they have experimental electrofishing for razor clams, which are much different methods. They use different waveforms, but really most of my research concerns the Dutch flatfish pulse trawling. And so here we have these electrodes which run parallel to each other, and so instead of trying to dig the flatfish out of the sediment, it causes them to kind of cramp up into a U-shaped form. The normal method for Dover sole to escape is to dig into the sediment, and so it prevents them from doing this. It causes them to become immobilized and to be caught into the oncoming net, which also is dragging along the bottom of the seabed. Here I just have a conceptual figure which I made to illustrate some of the ecological and biogeochemical effects of trawling. And so why is biogeochemistry important? Well, essentially it controls the existence of Earth's ecosystems. And just for example, most animals need oxygen to breathe and organic matter to eat. Plants create organic matter, but they need nutrients to do so. And trawling not only will remove benthos and other animals from the top of the sediment, but it can also cause lower water column oxygen, at least temporarily, with the sediment resuspension. It can also cause a lower denitrification and oxygen consumption from the seabed. And so this will mean more nitrogen being retained by the seabed and less oxygen being consumed. And to further illustrate some of this, here we can see that oxygen is naturally consumed into the sediments, while nutrients will tend to flux outwards. They can also flux inside, but they can mostly tend to flux out. These nutrients can lead to primary production in the water column, and as phytoplankton eventually settle on the seabed surface, we can use chlorophyllate as a proxy for this labile organic material created from primary production. We know that this is a high-quality food source for benthic microbes, and macrofauna alike, and trawling can certainly have a strong effect on these different dynamics. So, for example, it can lead to the direct release of more nutrients in the water column. We've also found out that it can lead to an increase in the oxic layer in the sediment. Now, this is also context dependent, because if you have more bioirrigators inside the sediment, these are animals which pull water and oxygen into the sediment, then they essentially increase the oxic layer. And if trawling leads to a decrease in these animals, then it can have an opposite effect. There will be actually a smaller oxic layer. Yeah. So this first chapter, it's titled Acute Impacts of Bottom Trawl Gears on Benthic Metabolism and Nutrient Cycling. It's a field study which we conducted in the Frisian Front area of the North Sea. And so we worked with professional beam trawl fishermen to create a tickler chain trawling site, and we did the same with pulse trawl fishermen, and we also had an untrawled reference area, but we took samples both before and after trawling as well. Most of these samples were collected with a box core, which collected intact sediment samples to gain information like chlorophyll A from the sediment surface. We subsampled these with incubation cores to gain information for things like oxygen and nutrient fluxes. And we also used these in situ benthic landers. Now, these were deployed on top of the seabed, and they collected much of the same information as the incubations, looking at things like oxygen and nutrients. We have here some data from the water column, and this is just for pulse trawling. Unfortunately, we were not able to do this for beam trawling, but we could see that here that every time the pulse trawler made a pass through its designated area, it created turbidity, and this turbidity was directly linked with oxygen, and this was very much correlated with each other. Now, for chlorophyll A on the seabed surface, we could see here the control samples, which had fairly high amount of chlorophyll A. After tickler chain beam trawling, we had about an 83% decrease in this, which was quite consistent. And we also had a significant decrease after pulse trawling, but it was a little bit reduced, as you can see here, and a bit more variable as well. Oxygen consumption also had similar types of, similar patterns here. There was less oxygen consumption after both types of trawling, significantly less, but a bit more consistency here after beam trawling. Here we have some data on oxygen profiles in the sediment. In control samples, these penetrated about 2 millimeters in the sediment, the oxygen. This was almost twice as deep after tickler chain beam trawling, and this was also a little bit deeper, but certainly not as consistent after pulse trawling. And so this data showed us that, well, suggested anyways, that there's a bit less biological activity from these sediments. This could mean that there's less benthic microbes in the sediment because they're more concentrated on the seabed surface, and other studies have found this as well. But before this, I don't think there were any other studies which have found things on oxygen consumption, this type of an effect anyways. So this next chapter is from the same research campaign. Now, unfortunately, this research campaign was actually plagued with all kinds of logistic issues. We had a big storm right in the middle of the campaign, and so, unfortunately, this study was mostly for beam trawling. We do have a little bit of a pulse trawl comparison, but we couldn't really do anything about that. It's mostly a beam trawl study, and it was more biological, more oriented to biology, the information from this. And so we see here a side scan sonar image from the beam trawled area. This area was clearly trawled in the middle, and you can also see some of the benthic landers from the previous study within this area. These are sediment profile imagery photos. The top row shows undisturbed sediment. There's a bit of topography with these, which is clearly seen. And in the bottom row, we have beam trawled images, and these are all flattened, as you can see here. And there's also this thin layer of fine sediments which occurred on the sediment surface. This is very consistent amongst our spy camera images. We also did HD camera transects, both in the beam trawled area and in untrawled areas. And so the most obvious finding that we saw here is this drastic decrease in epibenthos between untrawled controlled samples and samples which were beam trawled about 27 hours afterwards. And so here we actually found a 94% decrease in epibenthos in between the untrawled and trawled samples. There is a little bit of recovery from samples taken 50 hours after beam trawling, but not so much. Also, we looked at the burrow hole densities, which did see a significant decrease after trawling as well, but these actually showed a bit of recovery. And so this suggests that these animals may have still been inside the burrows. They weren't necessarily taken away but they had to go back to the surface and rebuild their burrow entrances. Here we have some output from our multivariate analysis. This is a between-class analysis, and the way to interpret this is here on the left side of panel A, we have our T0 and P0 samples. This means that they are untrawled. Next to those, we have our P1 samples, which are recently pulse-trawled samples. Above those are recently tickler-trawled samples, and to the right, we have samples taken about 70 hours after tickler chain beam trawling. And on the panel on the right here, we have a bunch of different taxa, mostly pointed to the left. And so you could pretty much overlay this graph over here. And this means that most of these taxa were seen in untrawled samples. And so it makes sense. But the interesting thing here that we see is that we have this species, Colleenassa subterranea, on the right. Now, why is it pointing to the right? It means that it was found more in trawled samples than in untrawled samples. Now, you sometimes get this type of an effect when you have benthic scavengers, mobile scavengers in particular, because they go into trawled areas to eat all the dead benthos which have been kind of dug out of the sediment. I don't think this is happening here because Colleenassa, from what I understand, is more of a deposit feeder. They just sift through the sediment and eat the organic material in there. And so what I actually think is happening here is that these animals are actually moving up in the sediment. And so they're doing this because they need to rebuild their burrow entrances. These are really deep dwelling organisms. They can go about 80 centimeters in the sediment. However, the cores that we have over here are actually about only 15 centimeters in. And so we didn't really catch these animals before, but after trawling, we had found much more of them because I think they had to rebuild their burrow entrances. And so they had to go closer to the surface, possibly leaving them more vulnerable to predation. And so people are always curious, of course, with the pulse and beam trawl comparison. And in this study in particular, we don't actually see much of a difference, or I don't interpret there to be a real large difference in between beam trawling and pulse trawling, just in terms of taxon abundances. Here, I don't think the difference, I think they both significantly affected the numbers of invertebrates, but couldn't find a difference for this particular study. So chapter 4 is a little bit different than the other chapters, which are more so field studies. This is kind of more of a laboratory-based study. We took samples from 9 different areas in the North Sea here and 2 different areas in the Dutch Eastern Skeld and subjected them to either electric disturbances or mechanical disturbances. And so the thought here is to try to separate and isolate the effects of pulse trawling, because pulse trawling can also create physical effects and electrical effects. And on the left-hand side here, we could see our electrical setup, whereas on the right-hand side represents our physical disturbance treatments. We used a laboratory pulse generator to create a homogeneous electrical field inside both the sediment and the overlying water. We did both a short-term and a long-term exposure treatments. On the upper left here, we could see the 2 different waveforms that we chose to use for this study. We have this here, which is PDC, and this stands for pulsed direct current. And so, in particular, this type of current only had unidirectional pulses, so the pulses only went in one direction. And this was a high-frequency current, and so this was about 80 pulses a second. Underneath that, we have PBC, which stands for pulsed bipolar current. And this is actually the waveform that is used in the flatfish pulse trawl fishery. Here we have positive and negative pulses, and this cycle repeats about 40 times a second. I mean, they could also increase and decrease this, but this is around that frequency. And they use this positive and negative pulses because they don't want electrolysis to occur. Because what happens with electrolysis is the electrodes will actually become degraded over time, and they don't want this. On the right-hand side, we could see with the sediment resuspension experiments that we mixed this top 3 centimeters of sediment, created resuspension, and then we collected water samples from the overlying water. We also did this with the post-drawl, the electricity treatments. Let's see. Here's an example of the physical disturbance treatment, the mixing treatment. We can see nutrients over time, in this case silica. Here, where this kind of purple marker is the disturbance treatment, and so we mixed the sediment, caused sediment resuspension, and silica was released into the overlying water. Makes sense. On the right-hand side, we have all of the data put together from the 11 different sites. We have our control samples on the very left in white, the electrical treatments in gray, and the mechanical mixing treatment in the red here. And so we could see that after mechanical mixing, you have this instant drop in oxygen here. Now this was almost consistent for all of our samples. We also had increases of ammonium, phosphates, and silica. For ammonium, we had an interesting effect in the springtime. The samples had a really limited release of ammonium, and this was regardless of how much organic material was actually in the sediments, or chlorophyll A for that matter. And in the autumn time, and so the samples were taken either in the spring or in the autumn, and in the autumn they almost always had a release of ammonium, certainly a temporal effect that we had here. And the only electrical treatment that actually caused anything was this pulse direct current for 120 seconds. I don't know if you can see that, but we had a short treatment, 3 seconds, a longer one for 120 seconds, and we found that after doing this electrical treatment, there was less phosphates in the overlying water. Now, why is that? Here we could see 3 cores, experimental cores, which were subjected to this pulse direct current, this high frequency current, versus a control core on the right. And you can easily see that the top of the sediment has this reddish brown color on the surface. And these, when you take a closer look, are actually iron oxides. And so what happened in this experiment is these iron oxides actually moved towards the sediment surface because of something called Ionic Drift. It's a way the electricity causes a directional movement for pore water ions. And so what this did when the iron moved to the sediment surface and reacted with the oxygen is it created rust, basically. And this iron oxide binds to phosphates. And so it consumed a bunch of phosphates. It also decreased pH in the overlying water. And I thought this was super cool, but really in terms of flatfish pulse fishing, we didn't see much of an effect for that at all with the pulse bipolar treatment. It's only if you change it into a high frequency pulse direct current that you see something. Here in chapter 5, it's much like chapters 2 and 3. We did this big field experiment, worked with a professional fisherman for this, but it's in a much different area than the Frisian front. And so the Frisian front, compared to this area, is actually relatively static. Here we went to an area called the Vlak de Van, which is about, the area we chose is about 10 kilometers off of the Dutch coast. So it's a coastal area, a lot more water coming in and out of here, so it's a high energy site. And we also knew beforehand that it's much more variable in terms of its macrofauna than a lot of other places. And so because of this, we designed, and when I say we, I really mean Gert van Rooyen, Joachim de Pestel, helped design this experiment to have not only one treatment site for pulse trawling and one for beam trawling, but actually 3 sites for each type of trawling. And so we actually did a treatment with the electricity turned on for pulse trawls, also with the electricity turned off, and of course the Tickler Jane beam trawling. We also had 3 different areas which were never trawled, these reference sites, and we collected samples before and after trawling. And so this study actually combined a bunch of data from all the same types of methodologies from chapters 2 and 3, as well as a bunch of other stuff, and so it's quite comprehensive. Here we have some data from a multi-beam sonar, and this shows the depth differences in between the different treatments, pulse off, pulse on, and tickler, in between before trawling and after trawling, T0 and T1. And you can see that there's just a lot of variability in all this data. And so, on average, we found about a 1 centimeter difference between before and after trawling for all these different treatments. Very hard to get statistical significance here because, though, of all this variability that we see. We also found that almost all of the samples were populated with linicid tube worms, sometimes in super high abundances, like you can see here. We also saw from the sediment profile images, you could see the type of bioirrigation effect that these linicid tube worms have. These dark areas are areas of anoxic sediment, and the tube worms will be pulling oxygen and water into the sediment, oxidizing this and creating this lighter colored sediment. It has really strong effects on sediment biogeochemistry. And on the right-hand side, this panel C, you can see this clearly trawled image. And you can tell because if you look closely, you have these tubes, and they're all flattened, all in one direction. Also, the sediment itself is mixed. You can see kind of a mix in between this oxidized and anoxic sediment here. Here we have some data on sediment oxygen consumption with this left panel, and Macrobentos densities on the right over here, in between all the different treatments, pulse on, pulse off, tickler, as well as our reference areas, and before trawling, after trawling, this is T0 and T1. For sediment oxygen consumption, we did see a significant effect for the pulse-on treatment, which actually had the most tube worms in the sample, and also with the tickler treatment. But it certainly was a bit of a larger effect with the tickler treatment. Not so big of an effect with the pulse-off treatment. Interesting. On the right-hand side, we have the Macrobentos results, and here the only significant result that we had was in between T0 and T1 for the tickler chain treatment. This graph represents pre-trawled samples on the left-hand side and trawled samples on the right, and we're looking at the relationship here between linicid tube worms and oxygen consumption on the top, and Macrobentos densities other than linicid tube worms on the bottom. And you can see that the correlation between linicid biomass versus oxygen consumption is really quite high in these pre-trawled samples. However, after you trawl them, it does not scale linearly, and this effect goes away. On the bottom, you see these Macrobentos densities, and so linicid tube worm densities are definitely correlated with the densities of other macrofauna, but this also goes away after they've been trawled. It doesn't scale linearly. And so this suggests that that trawling will kind of decouple these macrofauna faunal-mediated processes. This is also true for a number of other physical, biogeochemical, and biological parameters. And so in this general discussion, I talk a little bit about how this project is kind of in the middle of applied versus fundamental research. And so we have these applied implications. It's because we can directly compare pulse trawls with beam trawls. We can see if there's a bigger effect or a smaller effect, and this can be quite useful for managers. And it's fundamental because this is still relatively new stuff. And so a lot of the work is actually still on the cutting edge. And so some of our conclusions regarding pulse trawling start with any direct biogeochemical response of pulse trawl induced electricity is either undetectable or very limited due to these pulse bipolar currents. We didn't really see anything in terms of chemical changes if you keep this bipolar current. We found that biogeochemical effects from pulse trawls are moderately reduced compared to beam trawls due to lower mechanical disturbance, and so they very much follow the patterns of mechanical disturbance. And pulse trawls are a bit lighter than beam trawls, so it kind of makes sense here. We also found that the effects of pulse trawls on benthic communities can sometimes be just as impactful as beam trawls. Probably not likely to be worse because they're a bit lighter, but we found in Chapter 3 that the effect can be sometimes just as bad, or just as impactful also. These effects are, however, more variable than that of beam trawling and can lead to a reduced effect on benthic communities in certain habitats. We found this a bit of a reduced effect from pulse trawls in Chapter 5. I also know Berflon and Meesters just published a study earlier this year which found a reduced effect on Macrobentos communities from pulse trawls versus beam trawls. But this is important. It's important to know that we're looking at things purely from a head-to-head comparison. We're just comparing pulse trawls versus beam trawls kind of right on the spot, and there are more things to take into account here. So this is a figure from Reinsdorf et al. On the left-hand side, we have tickler chain beam trawling from 2009 and 2010. These are swept area ratios. On the right, in the middle, we have pulse trawling between 2016 and 2017, and we have tickler chain beam trawling from that same year. Now you could tell easily that there was more fishing on the graph on the left. And the graphs on the right, 2016 is kind of one of the peak years for pulse trawling. But you can see that there's still less fishing here. Now, why is that? And this is because pulse trawling for a sole is more efficient than beam trawling. And so this means that the fishermen will go out and they'll fish and get their catch quota faster than you would with beam trawling, and so there's less time actually spent fishing here. And in my opinion, this is one of probably one of the biggest benefits for pulse trawling versus beam trawling for the benthic ecosystem. You can still argue that there are some areas that pulse trawlers are able to go to now that beam trawlers can't, and this may be an ecological issue on a local scale. But in the grand scheme of things, it definitely seems that pulse trawlers will fish less, and this is important. Now, it's important. Now, the thing is, the people, a lot of the different groups that are against pulse trawls are not actually arguing that beam trawls are better than pulse trawls. This isn't their position. They're mainly against all types of bottom trawls in general, and they're saying that pulse trawls are just another bottom trawl. And I mean, I don't necessarily agree with the argument saying that electricity has a really bad effect on benthic life. I think there is a lot of evidence against that, actually. But on a purely mechanical standpoint, the difference in between no trawling and pulse trawling is going to be much higher than pulse trawling versus beam trawling. You know, it still clearly creates significant effects on the benthic environment. And so I could definitely understand an argument coming from that perspective. But the point I'm trying to make here is that pulse trawling can be thought of as either a good or a bad thing, depending on your perspective. It's very much a matter of perspective. Now, besides all this pulse trawl drama, we've also discovered a few other things as well. We found that here are some of our fundamental conclusions. We found that bottom trawling can lower benthic oxygen consumption and organic matter mineralization rates and reallocates much of these processes to the water column. Now we know that this happens directly after trawling within the trawl tract, but it's interesting to see that there are several other studies that don't really find what we found here. They find kind of the opposite effect with increased organic materials sometimes and higher carbon mineralization rates. Now this is very interesting, but I don't necessarily disagree with these other findings. I do think we may be looking at different things though. And so I'm quite sure that if you look at the trawl tract after trawling, you will see what we see: lower oxygen consumption, less organic matter. However, on a regional scale, this might look a little bit different. And this is because perhaps there is in areas with lots of sediment resuspension, you get a lot of this organic material settling on the sediment surface. And so not necessarily directly in the trawl track, but you might get a lot of organic material getting concentrated on the surface of the sediment, and this can possibly increase organic matter mineralization rates. But this is highly dependent on things like hydrodynamics, sediment type, however much organic material is in these sediments. In the North Sea, I think the currents are probably too strong and the organic material will be blown elsewhere. But there is a lot more work that could be done on this topic, a lot more left to discover to see what's really happening. We also found that the release of nutrients from physical disturbance will vary depending on organic material, grain size, and the time of the year when the samples were taken. And so basically, if sediments are more biogeochemically active, if the oxygen consumption is higher, if there's lots of fresh organic material, the effect on these sediments is going to be bigger. And last, we find that the biogeochemical impacts of trawling on biogenic reef communities can be remarkably higher than in pure sedimentary habitats. We found this in chapter 5. Now, the last thing I want to talk about is this last section, the appendix, which I added to this thesis. It's called Outlook to the Future. It details part of a research proposal that we wrote partly to build upon some of the research which we've been working on these past few years. Now, the aim of this project is going to be to create a type of tool to help predict the effects of bottom trawl fishing. And hopefully, also, this can help lead to facilitate innovation in the fishing industry for more environmentally friendly fishing gears. And this is going to be more of a modeling type of project. We're going to try to create some biogeochemical maps and predictive models, but we're also going to be running experiments to help calibrate these models. So this project has actually been accepted. We'll be starting on it shortly, and I'm very excited to get it going. So this is pretty much all I have for you today. I've had so much help from colleagues, collaborators, supervisors, and students. Really, this has been quite a dream come true for me. I've wanted to be a marine biologist ever since I was a young boy. Here is a drawing I made when I was about 5 years old of becoming a marine biologist. And so definitely super grateful to be here and to get to this point. And thank you all for helping make this possible.
Okay, thank you, Justin, for this very clear and comprehensive presentation. I can say that already. So we are now ready for a question and answer session, in the sense that I will first invite the members of the jury to pose their questions, and you are supposed to answer those. I would like to start with Adrian. Adrian, could you follow the presentation?
Yes,
Okay.
for sure.
Okay. So
I hope that my questions come also through to the audience
yes. Yeah.
in Ghent. Let's hope the connections will work.
Yeah.
So thank you, Justin, for your very informative presentation. And I'm thinking about how to spend a few minutes I have for raising a couple of questions or discussion with you. So I think I'll start with the political fight between the Bloom NGO, who successfully campaigned in 2017, late 2017, early 2018 against gulls fishing, and they were able to influence the decision in the European Parliament, which then voted for a ban on gulls fishing. And one of the main arguments was that the whole marine life would be electrocuted due to this gulls fishing. So my first question would be, if you look at the whole political debate, what is then the most important result from your study which can contribute to the debate?
Well, I think I could understand arguments against pulse trawling if they're coming from a point that that they're against all types of bottom trawling, and pulse trawling is another type of bottom trawl. I could certainly see that. What I have seen these past few years of doing research with the electricity, and we've also, I mean, you know, Adrian, that we've also done a lot of research on benthic invertebrates that I unfortunately was not able to put in this talk or in my thesis. And other people, lots of other people have done work on benthic invertebrates as well, also with fish and how electricity affects these animals. And I mean, from what I know, I don't think there has been any direct mortality from the electricity itself. I think that, and I'm not the biggest expert on this because this isn't part of my research, but from what I understand, the larger the animal is, the more it's going to be affected by the electricity. And so it's less likely that little worms, for instance, are going to be affected as much. The one animal, of course, which is in danger of having these adverse impacts is cod, which is a very muscular fish, and people have found that they can break their spines because they cramp up so hard that their muscles will break their own spines. Other than this, though, I mean, all the cod are caught in the catch process anyways, and they will probably end up on the dinner table. And so the consequences on the overall ecology, I think, are not going to be any different. I do not agree with the arguments saying that the electricity itself are what is going to cause these destructive effects on the marine environment. I think there's a lot of evidence against this. And so does that answer your question, Adrian?
Yeah, Joe, I think I fully agree with you, so I didn't really intended you to answer questions of the effect on marine organisms, with the things which basically your PhD colleague Pim Bouten has been working on in Wageningen. But I think a crucial result in your PhD is the experiments described in chapter 4, where you compare the direct effect of electrical exposure on these biochemical processes in the sediment with the effect of the mixing, just mirroring the effect of the bottom trawl disturbing the top layer of the sediment. And I think this is a real critical experiment which for me convincingly show that there is no reason for any concern about electrical effects used in the pulse fishing technique for sole. So I think that is really a major achievement and a major contribution for science-based decision making. I think that's really really important. And I think a follow-up question is that you used in your chapter 4, you did these experiments with many different cores taken from different locations in the North Sea, and you found that the effect of the mechanical disturbance was different between these cores. And I'm just curious to know from you, would this be an approach which you could use to estimate parameters which are required for upscaling effects of the mechanical effects of bottom trawling in an area like the North Sea? How do you do you think this type of experiments can be used in that context, and how could it be used?
Yeah, well, we could certainly try. I think these experiments show that in certain sediments, you're going to have an absolute increase of X amount of nutrients. And the thing is, though, this differs between the time of the year and the organic material in these sediments, and we definitely found strong correlations for those. But I think we could try to do an exercise where we try to scale up the possible effects of trawling by taking a look at these absolute increases in nutrients or decreases in oxygen, for instance.
But but then how can you translate the mechanical disturbance imposed in the experiment to the mechanical disturbance of all these various bottom troll types working in the on the North Sea grounds?
Well, we try to use per unit sediment units here. When I used two different types of units. I used the direct absolute changes in solutes, but I also converted them to meters squared. And so possibly we could use that to say that if you disturb this type of sediment in this area of the North Sea, you may release this many X amount of nutrients per meter squared. And so we haven't tried that yet, but that is something perhaps that we can try to do.
Yeah, but then the point of the depth of disturbance of bottom troll gears, which differs between the different gears, how would that come into play here?
Yeah, that is definitely an important variable. To be really accurate with this, we would probably have to use different depths. Of course, in soft sediments, the depth of disturbance is going to be much larger. With heavier gears and with different types of gears, you're also going to get a bit of variability in this. And so perhaps it's a good idea to conduct an experiment with disturbing different, kind of trying to create more of a gradient here. If you disturb the top 1 centimeter, you're going to cause this. If you disturb deeper layers of the sediment, you're going to cause something else. So, yes, it's not. I think there's possibly more work to do rather than just information that is in Chapter 4 to really try to create a realistic upscaling of this. But we can always try with the data that we have. It would be very limited though to whatever penetrates the sediment 3 centimeters, I guess.
Yeah. Okay, well, I can see that if you say elaborate the experiments in Chapter 4 by taking account of the different depth of disturbance, so the different layers, that definitely would, in my view, be an important step forward, which then could also be linked to experiments you are proposing and which you are going to do in the Outlook project, as I understand, where you are using the flume tank in Stellendam to tow gears. And I would like to make this link. So how do you think you can most efficiently organize these experiments to be able to give you a versatile tool which is able to deal with all sorts of different fishing gears?
Well, these experiments that you were talking about are more so looking at different factors affecting the penetration depth, actually. I think in terms of trying to do experiments where we look at the nutrient release in Stellendam with these, in the tanks there, I think it would be a bit difficult because we would need to have, we could always get a whole lot of sediment in there with a given amount of organic material, but the effects are going to change depending on, the time of the year or the amount of organic material, the grain sizes which are in these sediments. And so I think for those particular experiments, we were just going to look at the penetration depths and what, if there's something we could scale up from this, depending on using different fishing gears, actually.
But how do you? I think that's fully. How to say that? That's the real way to go forward, to look at the penetration depth. But then, how do you foresee to generate information which can be applied to different fishing gears? Because you can't tow all different relevant fishing gears in your project.
Yes, that's true. We're going to try to be as representative as possible. I haven't started getting so much into it just yet. Been trying to finish the PhD first before that. So, I mean, this is something that's still in the works. I mean, I have definitely been discussing a little bit with the people in Stellenbosch. Of course, there are certain issues. For instance, the gears do not, let's say we did an otter trawl type of gear, certain parts of the sediment are going to be much more affected than other parts. And so we're going to have to reconcile that by either looking at the average effect, average penetration depth for this. I mean, it's true for beam trawling and pulse trawling, of course, as well, because there's certain components of the gears which are going to be deeper penetrating than others. And so this is something that we are thinking about, but we haven't yet came to a conclusion for it. I mean, we did talk about trying to get an average effect across the whole gear.
Okay.
Yeah.
Adrian?
Yeah.
I would like to finalize this part of the questions. I don't know if you have a final urgent small question. If not, I would like to give the chance to the other members of the jury.
Now, I have another question, but that would take a few minutes, so that's maybe then better to do it hopefully sometime if we can have a beer together, Justin. Okay, thank you.
That would be nice, yeah. I have to apologize because I didn't introduce you, Adriaan. So we had the honor to have Professor Adriaan Rijnsdorp from Wageningen University, who was at the start more concerned about technical issues than about introducing you properly. So thank you, Adriaan. So we move now to the next member of the jury, and I would like to invite Dr. Engineer Hans Pollet from the Research Institute for Agriculture, Fisheries and Food, in short, info, at Ostend, Belgium, for posing the question. You can sit here and keep in mind.
Justin, thank you very much for this very interesting work. Thank you for contributing to the most studied fishing method in the world. I think you deserve quotation in the Guinness Book of Records now. And I would like to start with a general question. As you already said, a lot of things has been said on close trawling in the media, on social media, and even in the European Parliament. Many of these things were, I think we can classify them as fake news. False information was presented. So my question is, according to you, what is the role of scientists in this debate? Should we actively try to counteract this information, or should we stay behind the scenes? And I also ask this question in the light of the corona crisis and the experts we see on television, and there's also a whole debate around that.
This could be answered in many ways, I think. I've definitely heard different opinions of this. So our approach, of course, was try to not really get involved with politics, but I definitely have talked to other people and other scientists working for NGOs, for example, who say that why not get involved in this, because we know the topic much more so than other people. And so our scientists' role in this, I think, will really depend on the preference of the person. I mean,
did you feel the urge to respond more than you did?
Sometimes I did. I mean, at the same time, I don't know how big of an effect it would really cause. You know, I don't know how much weight our opinions of this will really create in the parliament. But I think it's good to know. I think people need to know this stuff. People should know what is real news, I guess, and fake news. At the same time, I don't want to fall into the trap of being biased either for or against this method. I think that in itself would be bad for the science. And so I would like to try to remain as unbiased as possible and just report what we find. And so perhaps the real answer would be to not necessarily advocate for it, but communicate. And so communicate the research. This is what we found. you know, we found a larger effect of pulse draws, or we found a smaller effect of pulse draws, whatever. And then the policy makers can take the facts then and do what they and make decisions based on it.
I tend to agree with that. But a few small questions. You were indeed plagued with a storm, and you decided not to use information from after the storm. Well, what made you decide that this storm actually had an effect? Did you observe effects, or was it more a theoretical approach?
I mean, we did observe some effects of the storm. We're actually hopefully eventually writing up a paper on the storm as well to see storm effects. And so that's really exciting, but it's kind of on the back end at the moment. I think at this point, a lot of it, we definitely saw effects in the water column. So, for example, I showed you this graph of pulse draw-induced resuspension and oxygen associated with that. And we also found information, we left it out there for the storm as well. And we found a lot of sediment resuspension with the storm.
At the seafloor level?
I think the probe was about 5 meters above the bottom of the seafloor. I'm not sure off the top of my head, but I think around 4 or 5 meters.
And the average depth was there 30, 35 meters or something?
About, yeah, that's right, 30, 35 meters. And so and we also actually had probes along, we also had some above that level as well, which measured turbidity but not oxygen. So yeah, we saw really big effects from the storm in the water column for sure. I think from what I remember, we kind of saw minor effects of the storm on things like oxygen consumption. I did see a little bit of a decrease with the untrawled samples before the storm and samples after the storm in terms of the sediment chlorophyllate. However, I don't think this is significant, and I think the trawling actually took off a lot more chlorophyllate than the storm did, but the effect is going to be much more large and diffuse, of course.
Okay, thank you. On page 56, you say that the primary cause of lower oxygen levels in the water after trawling is caused by the pore water and by the resuspension of reduced substances. Pore water, you really think has an effect on oxygen, given the big mass of water of the seafloor?
Yes, if it's resuspended, if you take especially the reduced substances within the pore water and you resuspend this into the overlying water, as these substances become oxidized, this will consume oxygen. And so this is just another thing that...
Okay, but then you're probably talking about the organic matter and not so much about the actual pore waters that has a lower oxygen.
Well, I think within the pore water as well, because in a lot of places, oxygen will only enter about a few millimeters deep. And if you have a deeper penetration of fishing gears there, then you'll get a release of a lot of material that is anoxic or hypoxic. And so this will carry a lot of things like reduced iron substances, for example. And as these get into the overlying water, they will react with water and oxygen, and it's another process which consumes oxygen. I think it depends on what area, how big this effect is going to be. But,
okay, fine. And on page 61, you state that bottom trawling resuspends organic matter, which can have consequences for benthic life and climate change. I think we agree with that. But I guess some people may argue that in the southern North Sea, where pulse trawling has been going on, it's quite shallow. There's quite a strong effect of storms, indeed, especially in the shallow waters. There's strong currents. So probably also the amount of organic matter constantly in the sediment will be lower. So some people may argue that pulse trawling compared to the other effects is probably negligible, so we can just go on. How would you respond to that?
it certainly could be. This is a big unknown. You know, I mean, it's theoretically, perhaps with all the bottom trawling occurring in the world, that it certainly could have an effect on the release of nutrients, and certain areas, for instance, are going to be at greater risk than others. Areas which, for example, in the North Sea, there are areas which have a seasonal hypoxia, And so if you trawl an area with seasonal hypoxia, I mean, perhaps it will trigger hypoxic events, and we know that through climate change, this is going to be more and more common, and so you can certainly argue this. I mean, there's a lot more work that we need to do on this topic. I mean, it is true that these big storms are going to resuspend lots of the sediment, of course, but I don't know how deep in the sediment the storm is really going to blow. It is going to cause these the sediment to come out, I guess.
So, I understand well, we don't really have a strong argument if someone when it says so as well goes from in the southern North Sea. Doesn't really make a difference on the general level.
On these larger scale dynamics, I think there is more work to do in order to be more confident in our answers for this. I think, I mean, some of the work we're doing right now, so for instance, we did the experiment, and then we had the big storm, and we're trying to now try to write up something with the big storm, and we also already published something about the trawling. And some of the effects of the trawling were actually much bigger than that of the storm. Like, for instance, with the oxygen consumption, I think the penetration depth of the gears was much higher than the storm itself. And so the wave-induced mixing in that respect maybe had an effect on the superficial parts of the sediment, the top few millimeters or centimeters, perhaps, but it wasn't as deep as with trawling. And so, I mean, I can't really tell you right now. I don't think we have yet a strong argument, but that doesn't mean we will in the future.
Yeah. Okay.
A final question?
Final question. Yep. Well, then just Jochen de Pastel and colleagues, among myself, did a study on the Frisian front as well. Quite similar study, but yours was larger in terms of types of samples you took. We have been quite frustrated not really to find any difference in Benthic, or the effect on Benthic life between troll and untrolled, or between pulse and beam troll. You did find effects on Benthic life. So would you say that you were just lucky, or you did a better job?
I think you guys actually collected more samples than we did. You used the Benthic dredge out there, from what I understand. We did, our area was, I don't know how many kilometers away it was, but I think it was to the south, or to the south,
to the southwest, I think.
I mean, it was in the freezing front, but not quite the same area. And so the area that we went to was actually a little bit more silty, and the area that you guys had was a bit more sandy. And you can actually see it from the spy camera photos by comparing the two photos. I know Jochen and I have done that. We do see that the grain sizes, you can just tell that they're a little bit different. Perhaps that had something to do with it, that it was in an area that was populated with a different type of benthos assemblages. But yeah, I don't really know for sure. Okay, right so.
Thank you, Hans. I would like to invite the next member of the jury, Steven de Graaf, from Royal Belgian Institute in Natural Science in Brussels, Belgium, but also visiting professor at Ghent University.
This is a special meeting. Well, first of all, let me congratulate you, Justin. I think that the presentation just gave was just great. And the study was great as well. What I particularly liked about the study is that there's a lot of things that have been said about post-trolling, post-trolling again, trolling as a whole. But what I very much appreciate is that this is really about, this is going beyond the impact on fish, beyond the impacts on stocks, on the stock dynamics, and or damage to commercial species, that sort of thing that are taken care of very well so far. But it is about the impacts on ecosystem structure and function. There's not that much. I think that's quite important, because fisheries management is not only about commercial species; it's also about the environment. So I appreciate very much that you contributed to that aspect. I have to put on my glasses. I had it in a larger font size, but even now I can't. My apologies for that. Yes. Okay. Let me just come back to what Adrian just said. I think it was his first question where he was talking about, okay, well, that the likelihoods that a pulse troll would actually cause major damage to the environment. You have demonstrated that this is not the case, and this is more or less the answer to. Yeah.
I don't know. I don't know if I agree with that actually. I think pulse trolls can create significant damage to ecosystems. I think any bottom trolls can.
Yeah.
But here our objective was to compare pulse trolls and beam trolls.
Yeah.
And from that, I think there is a reduced effect of pulse trolls. But I mean, even chapter 3 showed that it's very similar effects to beam trolls in that chapter.
Yeah, and that's exactly what I wanted to ask for. I have read it in these. Maybe Darren may have missed it, but if you look at the design of these trolls and the way they work, the way they are kind of moved to the environment as the job they have to call catching fish. Now, how could you possibly technically explain that indeed this impact onto, say, the benthic communities, the informal communities, isn't that big as we would expect?
How can we explain that it isn't as big as we would expect?
Yes. Yeah.
I think it's good to put things into context with other bottom trawl gears or bottom dredging gears. We have already trawled the North Sea a lot with beam trawls and otter trawls and dredges and different things. And so, in order to put things into context, of course, I think we need to provide proper comparisons. And so, with pulse trawling, I think they can still create a significant impact, but on average, it's going to be more variable than that of beam trawling, and it's going to be, on average, probably reduced. And I think the biggest effect of pulse trawls, or the biggest benefit, is actually that they fish a bit less than beam trawling does. But I think any type of bottom trawl gear will create that impact. Yeah, sorry, I don't know.
Yes, but I was looking for the technical explanation. If you look at the technical design of a pulse troll, then actually, you do not have the stickler chain, so actually the bottom should be less reworked than in comparison with the classic beam troll.
Yeah.
But still, you do not find a major positive or less negative effect of the pulse troll.
In chapter 3 we did not, but in chapter 5 we did. And so I think it depends on the benthic assemblage, on the environment as well. I would argue that in soft sediments, perhaps like in the Frisian front, the sediments are going to be more easily penetrated by bottom trawl gears. I also know that Macrobentos and Eric Meesters published a study earlier this year where they actually found a reduced effect from pulse trawls compared to beam trawls. I don't actually think it was significant, but I think it was reduced. And so, I mean, we have a little bit of variability in the results. I mean, I have one study that shows that there's a reduced effect, the other study that does not, and then we have Berkman and Meesters, which does. Yeah. I mean, I don't actually think the effect is so much reduced. I think it makes sense that it's reduced because it's lighter. But I mean, a bottom troll, I think there's going to be a level of disturbance using any type of bottom troll, really.
Yeah, that's clear. But I would have expected a more significant difference between both types of activity. That's how it is. And then just referring to what you just said, indeed there must be a difference between the different types of environments. Right. And so you have done a study in the Frisian front. Frisian front is, of course, a very nice environment to investigate when it comes to fauna. Well, there's a rich fauna community, a rich benthic community over there, so it is very interesting, very exciting to do some work over there. But how representative then is that study compared to the North Sea environment? Frontal systems are not the most common systems throughout.
Yeah, that's true.
The world's marine environment.
Yeah.
So then you opt for like an atypical system to do your research, to learn something about the impacts of different types of fisheries. So how relevant is that?
That's a good point. I mean, the Frisian front certainly is a unique system. I mean, part of the reason why we chose this system is because we know a lot about it. There's a long history research there, and so we thought we could better relate it to previous studies. But indeed, it is unique in the respect that it is a frontal system, only representative of certain North Sea habitats, and so yes, indeed, I think it probably is a little bit limited, and so we may not try to extrapolate it and say that this is something which is happening everywhere.
Mhm.
And so I did try to, I know you made a comment about that,
Yes.
and I did try to add that in somewhere, that it's not going to be representative of a lot of different other habitats, but I mean, it also depends as well. There are certain smaller types of habitats, let's say off the Belgian coast, which are filled with loads of organic material, cable bacteria, and different things like this. And I mean, these also are not going to be representative of other types of habitats. You could also say the same thing for our Chapter 5, our Leniche tube worm dominated habitat. And so, yeah, I guess, I mean, it's difficult because we want to choose an area which is representative of other areas, you know, but we also want to choose interesting areas.
Of course. No, I thought that there may have been, for example, a link between the target fish species in the fisheries and the environment that you targeted with your research. I think that when right, the coast trawlers they specifically target, is it sole? It's flatfish, but is it sole?
So, right. Yeah, I'm not. I mean, I'm not the biggest expert on this, but from what I understand, I don't know if they actually catch place better than other beam trawlers. I think it's really primarily sole.
Okay.
Because the sole they bury in the seabed like this,
Yeah.
and then they're very flexible and they're able to cramp up real quick, and this is really where the false stalls excel in catching.
So I'm not a fish ecologist, but I may guess that sole may be typically found in that sort of environments as well. They definitely are. They may be found also beyond that kind of habitat, but I think that the frontal system, well, or the benthic system, like we find in the Frisian front, but also in front of the Oops. So it was just in front of the Belgian coastline, where we find our sole and where sole fisheries take place. So from that perspective, it may be more representative than we think.
Yeah, I mean, we saw them out there. I mean, with our video cameras as well, we did see sole in those grounds. I don't know how many. I didn't think we counted them actually, but they were there. So.
Okay. Yeah. Thank you. Yes. Thank you very much.
Okay, thank you, Steve.
Okay, thank you, David. Then I would like to invite Ulrike Bergman, Dr. Ulrike Bergman. She's from Ghent University, Department of Biology.
Thank you. Thank you, Justin, indeed, for the very clear presentation. While I was reading your thesis, I thought it was a very challenging topic, but you managed to formulate very clear conclusions. Although there are still things unclear, you came up with very clear conclusions that can be used. So I mentioned that I thought it was a very challenging topic because you were combining these field experiments. You had to deal with storms at sea, but you also took a holistic point of view. You tried to measure the Pelagic coupling. So you had measurements in the water column, measurements of the sediment, and you combined that with experiments in the lab, where you tried to simulate the process in cores. But I mean, so that's what I mean with it's challenging, it's difficult to simulate all of this. So I was wondering, that's my first question, how you came to to the idea to measure this all, well, the experimental process in the lab.
How we came up with that?
Yeah, and also why taking cores and measuring the responses in terms of biogeochemistry on board and not in situ, or not entirely in situ.
Yeah, well, a lot of thinking and years of planning. We had lots of ideas. We had ideas of fitting electricity on an electric pulse trawler, or not trawler, what do you call it? This electric pulse generator and wires hooked up to maybe a box score and electrifying the sediment and then collecting samples afterwards. But it was very impractical. And we also even made this. We even created this custom-made incubation core with so that we could put these flat electrodes on the side of them, but it definitely didn't work. And so we elected to choose the system that we did because we know that, for one, the electricity penetrates the sediment almost as much, well, I think actually just as much as it does the water. So if you have waterlogged sediment, it's going to penetrate it. We chose to use flat electrodes because it creates a homogeneous electric field. Now, we don't actually see this with pulse trawling in the field. We will have a stronger effect closer to the electrode. That's what we would really see. But then in terms of an experiment, it's going to make things very difficult if you're going to have one part of the sediment strongly affected and the other part of the sediment not. And so we elected to kind of create a bit of an overexposure of electricity on these sediments to see if there really was a response. I mean, we didn't know what was going to happen, really. And so this was part of the decision-making process. Also, I mean, it would have been nice to have the electrodes parallel and horizontally like this, but it's hard to have real flat electrodes that do that, and it's hard to have a sediment core and have the overlying water mixed homogenously. And so that's why we do things the way that we do.
So actually, you needed a chapter in the PhD of everything that you had thought about, but that didn't work.
Yeah, it's very challenging.
Yeah. But you, I think now in the final version, you had a scheme of a new design to measure the effects of the process on the activity of fauna. I think it was with Ammonium where you had the
Correct.
electrodes in parallel. So is that an improvement as opposed to the course where you send the pulses vertically?
I don't know if we would call it necessarily improvement because we're looking at different things here. We did not. So this is a study where I was looking at the effects of benthos and trying to. It's not included in the thesis, but we tried to see what the effects of electric pulses are on benthic functioning in terms of bioirrigation and behavior. And so we would see if the worm kept bioirrigating or if it would stop. And so we did not actually look at different nutrients in the sample. We looked mainly at oxygen to see if there was it would keep irrigating and oxygen kept going into the sediment, or if it would have stopped and had this for a prolonged period of time. I actually don't think it is an improvement to what we did in chapter 4 because we were measuring something completely different in that chapter. I wish I could have this finished for you so I could have it as a chapter because I'm still quite excited with that experiment. It's definitely really cool, but it's not there. So we're still working on it.
Yeah, and you formulated that as something that is that needs to be investigated in the future, specifically the effect of the pulse fishing on the endemic behavior.
Correct. Yes.
Yeah, that was actually part of my question. For chapter 5 on the pulse fishing and beam trawling effects on the Raniche reefs, I was actually wondering, it would have been nice to actually see what has happened there, because it was not entirely clear to me.
Yeah.
When you showed the picture, the sky pictures of the Pelagic tubes before, and then the sediment with the flattened tubes afterwards.
Do you want me to pull it up? Or?
It's fine. I think we can discuss like this. I was wondering where the lanishes had gone to. Did they draw deeper into the sediment, or were they were gone? Yeah. They were removed.
Correct. Yes.
And the currents had also swept them away.
I think the net.
The net.
Actually. Yeah. I know that just from talking with fishermen that they had trouble a lot with these Laniche reefs because they get tangled within the nets and cause problems with fishing. It's less efficient, and so they have to remove them. And so I've actually seen pictures of lots of Laniche caught in the nets. I don't know if it's necessarily the gear which catches them, but I think they get snagged into the net, and then the net carries them away. And so I think they were actually physically removed from the fishing and the worms inside of them as well. And also, it's flattened, you can see. And so there's less worms, and it's also flattened in one direction.
Yeah. So it's the lower sediment community oxygen consumption that you observe after trawling is then to be related to the lower biomass. I would have expected that if there was some dead biomass on top of it, that there would have been a higher sediment community oxygen consumption because of the bacterial degradation. But now that you're saying that the biomass was actually removed, okay, that makes sense.
Yes, I forget exactly. I don't think in terms of biomass we actually had statistically significant differences. Certainly we did for abundances, but yes, they were physically.
One final question is that what? So you at one point you write that upon trawling the organic matter is being suspended into the water column, so the sediment, the oxygen consumption is being replaced with the water column. So
Right.
there's less in the sediment. What consequence does this have for carbon cycling?
I mean, you can argue that it will reduce carbon cycling in the sediments itself. Of course, the carbon cycling in the sediment is going to be slower than in the overlying water because the overlying water is filled with oxygen. And so in the sediment, oftentimes you only have 1 or 2 millimeters of oxygen in there. And below that, it takes a lot longer for the carbon to become degraded with anoxic mineralization processes. So it's much slower. And so I think if you resuspend them into the overlying water, this will quickly mineralize a lot of the carbon and turn it into CO2, which gets released. I think much of it will really depend on how much organic material is in the sediment and other dynamics, but that's what I think will happen.
So in the end, also less carbon being burned.
Yeah, we've looked at this and talked about this. I know that, theoretically, I think yes, that can occur, but I know the amount of carbon which does get buried is always often very small percentage. And so I'm still a little bit hesitant on that. Theoretically, yes, it could happen, but I'm not sure yet. I would say it just has a bigger effect on mineralization processes that I'm a lot more confident about.
Okay, then I would like to invite the final member of the jury, Professor Tom Moes, also from this side of the Faculty Science at the University of Marine Biology.
Okay. Justin, first of all, I want to congratulate you. I think you presented an excellent piece of science. At least I can judge on the written book. I was very sorry to come in only at your last slide of your presentation. From the way you have been addressing questions, I have the impression that you may be even better in communicating face-to-face than in communicating on paper, even though the written work is very good as well. I much appreciated, like Stephen, that your view that the whole problem is not so much directed at a particular target species or whatever, but really at the ecosystem functioning and the underlying ecosystem structure. I think that is a very strong point. Now, as most of my questions, or maybe even all of my questions, have already been addressed in some way, I may be a little bit repetitive in what I'm going to put to you. And I would like to just start by the message that I thought I took from your last slide, which was a little bit diffuse in its message. If I would be in a position where I could take decisions. So suppose that I were the new Minister of Agriculture and Fisheries in some country that is about, well, that has taken a stand against pulse fisheries in the past, but that I want to take an open view. How are you going to summarize your main message to me that might influence what I'm going to do?
I think I'd probably say something like pulse trawling still creates significant effects on many ecosystems, but they're less than withdrawals.
And if I would then ask you how much less, I mean, is this an effect that makes a difference? What would be your answer?
If you do a head-to-head comparison, then it's moderately reduced, I would say. I think the main benefit of pulse trawls is the characteristic that you don't need to fish as long, and so there's less fishing in general in order to get your catch quotas. And so that, I think, is the main benefit of the pulse trawls. I did not actually do those studies myself. I know Adrian and Hans have been looking on this stuff, but that's I would refer to these things.
Then, as a beginning minister, I would probably have some doubts on how to enter my perhaps different view with at the back of my but at my back, let's say, a number of critical representatives of both environmental organizations, but definitely also the fisheries themselves. And Hans already probed after how should we communicate about this as scientists. So if I were to ask you, you as an expert, can you please send out a tweet that reopens the debate without taking a clear stand in favor of one or the other, but what element or elements could you in a brief message put forward that would reopen the debate?
I think I would focus on the effects of electricity, because from what I understand now, a lot of the decisions, I think, were made on the basis that electricity is causing a big effect on the benthos, that people have this idea that the electricity is killing all the fish and killing all the wildlife out there. And this is simply not happening. The fishermen want fresh fish, and so they don't want to be doing that. I mean, I'm not for nor against pulse trawls. I'm trying to take a neutral stance on it. I still think it creates significant effects, but not perhaps not as bad as beam trawling. And so it's difficult for me to really take a hardline stance, but I think if I were to say one thing that I wanted spread out for everyone, I think it would be this lack of an effect of electricity. Because most people just think that it's way, way worse than it actually is.
Yeah. Well, but that I think is one of the things that I appreciate a lot. I think as scientists we have to always try to be balanced in what we say, but at the same time it's more and more difficult to get through to a broader audience if you put more nuance in your story. So the things that you are sure about, like the lack of an effect or direct effect on the electricity, are things where we have to dare communicate more strongly. More openly. So yeah, I think that's a very strong point. And I suppose that I had some budget available for you. As a minister of agriculture and fisheries, what would be, let's say, a 2 or 3 year project? What would be the thing or things that you could investigate there, and in what way that could present a real breakthrough? Not necessarily a discussion between bean trough fisheries and pulse fisheries, but just anything that could make a breakthrough with less environmental impact from trawl fisheries in general.
Oh, well, that's a good question. But I think I would be doing what we are going to be trying to do already. With this appendix chapter, for instance, we just received, this was accepted, of course, a few months ago. And there's a lot that we still don't know about the effects of biogeochemistry, the trawling effects of biogeochemistry. And this could be something which is very important because it affects all benthic life out there. If things are going to lead to hypoxia, eutrophication, and things like that. So this could theoretically be something which can be important to the whole ecosystem. Yet we still do not know so much about it.
And would you recommend that as perhaps the most crucial criterion to assess impact of trawling? I mean, if it doesn't have a major impact on the biogeochemistry, we can say that it's more or less okay.
I don't know if I would say that. That's challenging. I think that we just don't know a lot about it, so it's just something left to discover. I think we should perhaps stick to, in that sense, do research on things that we do know about, so perhaps fish stocks, things that people find important. So I know don't really take. I don't know how many people are so interested in all the nutrients out there, but if you're trying to convince people of something, it's easy to say that. Oh, we look at fish populations, and everyone's interested in this.
It's like more tangible than the biochemistry for the fluoroids.
It's easy for people to get around. I mean, I guess I think biogeochemistry is very important, but I am realistic in the sense that we still just don't know a whole lot about it. It could theoretically be something which is very important, very interesting, but there's so many uncertainties that we just need to keep exploring and investigating this topic. Yeah, to get the public behind it,
Well, I just hope you have the opportunity next years to do the necessary fundamental science that is not biased by any prejudice, but at the same time also communication about those things so we have business. Thank you for your presentation and for your PhD.
Okay, so we are nearly at the end of this first part. Normally I would give the floor to the supervisors promoters, but it seems I will follow more the Dutch tradition and give them the word afterwards. It gives me, we are also a little bit short in time because we had really interesting discussions, but I still need to ask if there are questions from the public. So we have 2 publics here. I don't know if there were questions through the chat, but I can see already a hand of an expert in the room. So please, Jochen.
Thank you, Justin. Thank you for inviting me. I'm very glad I could be here, and I agree with the jury that you have a very nice presentation, really enjoyable. I also think it was very enjoyable to look at the interesting findings that you have on the acute impacts, and it's very innovative to see the studies on biogeochemistry, which is a really new thing. I'm very happy about this. And that's also where my question relates to. So you have studied the acute impacts now, and I wonder about actually the long-term effects. I know it's outside of studies that you've done, but it's just your outlook to the future. What do you expect, like particularly on the decreased oxygen and decreased carbon mineralization that you found in the acute study? What do you expect to see on the longer term? Is this something? What does it do to your ecosystem with the continuous fishing?
Yeah, well, I mean, we did see that the oxygen consumption and probably the carbon degradation is probably reduced at least for several days. I think it would depend on the amount of organic material which is deposited. And so this happened in June, for instance, and so it could perhaps be recovered quicker because you're going to have more organic material coming in again, and so the oxygen consumption may go back to its original level because of the deposition. If this happens in the wintertime, I think the recovery would perhaps take much longer because you're still waiting for that deposition of organic material. I mean, we have explored this a little bit with modeling, and we plan to do so more so with this upcoming project, which I talked about. The PhD, though, itself, I mean, it's very difficult to look into long-term effects sometimes because there's so many different variables and temporal things to take into account. But I would argue that it's going to be, it depends on when the organic material is going to come into the system.
Okay, thank you. No questions from the online audience? So then I would like to close this part of the event. Don't run away and stay with us. I would like to invite the jury to meet separate, and we come back soon. Announce that Justin Tiano has obtained his PhD in science, marine science at Ghent University, and that the judgment of the jury was based on of course, the excellent science that was provided through his work. He really gave an added value to the topic. The biochemical component was something where science was waiting for, and you just came in on the right time to give these insights which were really new to the field. We also have to congratulate you with the way you were communicating. It's obvious you're a strong communicator, and although you were very careful in how to translate your results into policy recommendations, maybe you should go into politics. You have all the skills and the brains, the way you communicate. We enjoyed your presentation because it was really entertaining. For specialists and a wider audience, you took us by the hand from the beginning, and we were in the story, and we enjoyed very much. So the embedding in the political context, although you kept your independence, you didn't want to make strong judgments, you only wanted to communicate clearly, and you did. So that was also very much appreciated. We thought that you also provided to-the-point answers to the questions. So at the end, only very positive appreciation for what you have been achieving over the 4 years, but especially today in presenting your work to us. So, Justin, we have the tradition to not only give you this piece of paper with your diploma, you also get this barret, which you may put on your head yourself. Maybe you're even more familiar how it goes, because it's actually an American tradition that we have borrowed. Yeah, we were talking about it, we were not sure. But it's for the picture. And then it has a tradition to provide you with these colorful socks. Wow. So we give you a bit of promoter from the Netherlands, because of course this is mainly work you have been doing at the Royal Institute of the Sea in Pelagic. And Carleen, I would like to give you the floor. Maybe you have to stand here.
Okay, so we do this because normally we have a reception where we can talk to you in private, and so now with Corona this is all not possible, so we thought that we were going to make like a combination of the Belgian and the Dutch staff of doctorals. So what I did is I prepared a laudatio that I will read now. Dear, wel edel zeer geleerde Justin Tiano, beste Justin. This very long title of wel edel zeer geleerde is the Dutch formal way of saying that you have successfully defended your PhD and can call yourself a doctor in science. As your promoter, I have been chosen to read your laudatio, which I will do with pleasure. Now, during the day of your interview, we had two vacancies. One to study underwater sand waves, and one to look at the biogeochemical impacts of two types of fishing here. For one reason that is not entirely clear, you applied for the other job, to study sand waves, although you said to be also interested in the fisheries subject. However, many candidates for the Pulse project were mainly interested in the fisheries itself, and not so much on the biogeochemical effects fishing has on the ecosystem. Also, many were self-declared vegetarians and or were strongly associated with environmental movements and sometimes even against fishing. I have nothing against vegetarians, but to oppose fishing is not a good starting point for a scientific exploration of their effects. And so we deemed this to be a risk for a job in which we needed to cooperate with fishermen. So during this interview, you showed to be highly passionate and well motivated in marine biology, but you also gave the impression to be open-minded and willing to take on new challenges. Another asset that you had was that you did not a priori oppose against fishing, and of course, you have great verbal skills that were already clear during the interview. So we decided to hire you, and you started in spring 2016. Your duties were to collect data on the biogeochemistry of the North Sea and perform small-scale experiments on board of our Resuspension, and to piece and as the pièce de résistance to perform two large-scale experiments with fishing boats dragging their gear on a small part of the sea bottom, after which we had to determine the effects on the biology and biogeochemistry. My God, this is very The first experiment in 2017 took place in the central part of the North Sea, the front. The other one was closer to shore in the Vlakwanera. As these field campaigns are quite risky, I also thought it was a good idea to do monthly sampling in the Oosterschelde to quantify the biochemistry there. So this as a means to learn the skills that you needed, and also as a safeguard in case the North Sea experiments would fail. So the first big event was fisheries experiment number 1 in the Phosphates. Except for Rob Wittbart and Magda Bergman, we were all newbies in this campaign. You as a fresh PhD student, and I had never participated in a scientific cruise on the North Sea, and yet I was to be the principal investigator. And the campaign started off horribly, with the fishermen not being able to come and perform the experimental fishing. But no worries, just before we set sail to the Friese front, you were able to find another fisherman that would do the experiment. There was one problem though, which was that this fisherman started 8 hours earlier than we had foreseen. So we had zero time to properly prepare this experiment. This is why we were left with very incomplete T0 samples. Also, as everything had to be crammed in a very short time frame, it also looked as if in the evening that we would not even get proper T1 samples, as we had already worked for more than 14 hours before we could take box scores. But then, fortunately, we were on the Pelagic, which has a very flexible group, and so we could convince them that these samples were really, really, really necessary, else the entire experiment would have been ruined. During the same cruise, there was to be the second fishing experiment with the pulse gear, 3 days after the first one, and also for this one bad luck story. We saw the day before the second fishing experiment that a storm with the intensity nearing a hurricane was ridiculous. Now this was really bad luck, as we had chosen the experiment to be done in summer, so as to be sure to have good weather. Well, we had plenty of time to prepare the day before the experiment, so this time we had to record a perfect zero situation. And the fishing occurred while the storm was getting worse. So we really try to do as much as possible after the fishing, but many of our plants had to be aborted soon after that, as it became way too dangerous to continue sampling at the box or even to be on deck of the Pelagic. So we had to abort our work and were forced to wait until the summer storm passed away, which took the remainder of that day and also the day after that. I must admit that the rough sea during the storm was absolutely beautiful and awesome, but the result was that we had done only a fraction of what we could have done and had planned for this time. Now, many students would feel completely depressed by these circumstances, but somehow we decided to call this an opportunity rather than a failure. And although many of the referees did complain about the experimental design not being a traditional Baki design, we were able to get 2 papers published from this experiment, one on the effect of fishing on biogeochemistry, one on the effects on the fauna. And there is still the data set that shows what happens to the bottom of the North Sea when such an occasional storm hits in summer, and that could be written down as a scientific paper. What I learned during this cruise about you is that you have a very professional scientific attitude. Your work on board was never compromised, even during our rush to complete all the tasks in 2 weeks' time. And this has been a constant during all the 4 years of your PhD. You work very effectively and very efficiently. You are very efficient in organizing things, and then you can do everything yourself, up to wrapping up your results in the form of a scientific paper or with a presentation either to the PhD community or for an international audience. It doesn't seem to matter to you. You are not afraid to work long hours and to get tasks finished. We even did a complete night campaign in the dark in the Westerschelde, where we looked at the migration of Macrobentos during the night and sampling for a complete tidal cycle, a novel data set that should and probably will be published. As a person, I think I can say that you're very easygoing, friendly, optimistic, and you seem to get along with everyone. Also, you're not afraid to engage in a discussion, be it with the press, with your peers, or with colleagues. If we have a meeting in Iersen and nobody asks a question after the presentation, then we know that you're not there. As you always ask a question, and usually there are good and general questions. I would like to end this introduction with your frustration, which is not being taken seriously enough as a PhD student. You had the impression that important things were decided by the management without consulting the PhDs. And now I will tell you a secret, sometimes we as senior scientists also have the same impression. But of course you took matters into your own hands, and before I realized what was happening, you had involved all my PhD students to take part in a commission that was to liaise between the PhD students at Iersen and management. So we headed this group and had regular meetings with class, the head of our department. And then 2 years ago, we made you participate in the selection of a PhD candidate for another project because you had requested to get such an opportunity. And of course, you actively participated in the selection procedure, but nevertheless, this experiment was not a big success, as apparently many PhD candidates tend to ignore PhD students during this period and focus only on senior science. So now you are no longer a PhD candidate, but a true doctor. I hope that in your future career, be it as a scientist or in management or wherever, you will remember what it feels like to be a PhD student and take all your future students very seriously. You will realize soon that PhD students still have a lot to learn when they start, but they also have an immense capacity to take on new challenges and learn new things. However, sometimes they go in unforeseen directions that may either end well or completely fail. So these are the tricky parts, as you will need then to try to guide the students in what you think is a good directions without giving them the impression that they are not taken seriously. I have only 2 regrets from our work together. One is that you never learned to make mechanistic notes, but I'm pretty sure that this will change in the coming 2 years, as now you will be forced to make notes and describe the effects of fisheries on biology and biogeochemistry. The second regret is that with your PhD thesis, we were not able to convince the members of the jury that it does not need to be all research chapters that need to be included in each PhD thesis. Because we decided to add also the project proposal that you maybe wrote, and of course I helped a little bit, and that was granted. Yet the jury decided that the scientific proposal, even successful, did not fit as a regular chapter in the PhD thesis. I think it's especially a lost opportunity, as nowadays science is more and more moving in the direction to have a broader evaluation of science, of scientists, and to look also at a more complete palette of skills rather than just scientific papers. And I still think that the well-written project proposal perfectly fits within these new assessments. I do remember, though, that it was a close call, and if Stephen McGrath would have been present during the defense, we would probably have been 50-50.
Yes.
Anyway, the good news about this is that your proposal was granted, so that you will be working for us on the fisheries impacts for the next 2 years at least, but now as a project. To finalize, I would like to congratulate you once more with this important step in your career. I know it means a lot to you, and so let me tell you that it also means a lot to many of us producers. So to celebrate that, we have a number of gifts. One is from EBS and the other one is from you, but I forgot.
Which one is it?
Yes.
Alright. Congratulations. Oh, cheers. Cool. Well, thanks, EBS.
There's more.
Oh, wow.
To enjoy?
Oh, wow. My God, I don't even know what's in here.
So what do you think? Okay. Keep it short.
Well, this has really been a dream of mine. I wasn't kidding when I brought that last slide up there. I mean, a lot of people, I hear all these horror stories about doing a PhD online, and everyone talks about, oh, it's the worst time of your life, all this. It was the exact opposite for me. I loved it, really. I've been wanting to do it for a while. I could have a better supervisor and co-supervisor and a place to work, really. It was so rewarding for me. And I would do it, for the rest of my life if I got paid enough to do it. I definitely would continue doing this work. And I'm so grateful for all of you. Thank you all for being here. And thanks for giving me this opportunity.
Okay, then we can close this session, and a pity we can't drink a glass on this special event, but I heard already people making appointments for having a beer together and discuss further this very interesting topic. So,
Okay.
thank you.